In 1995, Katalin Karikó had hit a low point. A biochemist at the University of Pennsylvania, she devoted her entire career to turning mRNA, one of the most important building blocks of life, into a whole new field of medicine. But it just wasn't happening.
Everybody argued that the RNA is so transient and degrade and it will never be medicine. I knew that it can be used for everything and, kind of a Cassandra feeling that I can see the future and nobody believes me.
Karikó's grant applications were rejected. She was demoted, endured a cancer scare, but she stuck with it. She spent weekends and holidays doing research and experiments, and in 2005, Karikó and her colleague Drew Weissman published a breakthrough study confirming the therapeutic use of mRNA, research which would underpin the success of mRNA COVID vaccines today.
When the first COVID-19 vaccine was approved, I was extremely excited. I thought it was a changed moment for mRNA. What we've been seeing in animals was real. It works in people, and that's the most important thing.
mRNA has gone from a rejected idea to a powerful weapon helping to end the pandemic, and applications of this technology could provide a cure for diseases beyond COVID. Messenger RNA, or mRNA, is a genetic material that copies instructions from our DNA. Our cells use these instructions to make proteins, which carry out different functions in the human body.
mRNAs are responsible for everything our body does, from breathing air to eating food to walking to sleeping to thinking.
COVID vaccines from Pfizer, BioNTech, and Moderna are based on this mRNA mechanism. Unlike traditional vaccines using dead or weakened viruses, mRNA vaccines use the genetic information of the virus. Scientists make an mRNA that codes for the spike protein of SARS-CoV-2, the virus that caused COVID-19. Then the mRNA is put inside a lipid coating and injected into the human body. The cell reads the mRNA and starts to make harmless spike proteins of its own, which triggers the immune system to produce antibodies against the virus.
The way that mRNA vaccines work are similar to really all vaccines. What they're doing is they're presenting a protein or proteins from a pathogen, and they're causing our bodies to make an immune response against those proteins or protein. When you make an inactivated virus vaccine, you have to isolate the virus, you have to grow it up in eggs, you have to figure out how to grow it in eggs, you have to purify it, inactivate it, and formulate it. With mRNA, you only need the sequence. You don't need the virus, so it's a very simple vaccine to make.
This means that making mRNA vaccines is faster than making traditional ones. But the journey for mRNA to break into mainstream medicine has been long and challenging. mRNA was first discovered in 1961, but it wasn't until 1984 that scientists were able to recreate it in the lab. In the 1990s, as scientists knew more about the makeup of human DNA, gene therapy became popular, inserting a gene into a patient's cells to treat or prevent disease. But most researchers gave little thought to using mRNA as a vaccine or a drug, despite its close relationship with DNA.
I argued that the messenger RNA is more like a conventional drug. You can apply, and then if you see beneficial effect, you can reapply, and then the healing is done, rather than a gene therapy, where you are delivering the gene and you cannot control.
But many rejected Karikó's idea, arguing mRNA therapies didn't look feasible. The immune system is hypervigilant against foreign RNA entering the body, and so the injected lab-made RNA caused inflammation, which destroyed it before it could start to trigger an immune response. And even worse, that inflammation could be life-threatening. In 1997, Karikó met Drew Weissman, a respected immunologist, and they started to collaborate.
The big discovery was we figured out by modifying the nucleoside. So RNA has 4 letters that code for a protein. Each letter is a nucleoside. or a base. We modified those bases, and that reduced and got rid of the inflammation.
When we altered uridine and we had pseudouridine, we found that not only we have RNA, which is non-immunogenic, but we had very high level of protein production.
The amount of protein made from the RNA increases about 1,000-fold. You get rid of inflammation and you increase potency in the same step.
So it was like a dream come true.
They published the key research and filed a patent, which was later licensed to Moderna and BioNTech, the companies making mRNA COVID vaccines today. Carico also started to work for BioNTech in 2013. But there was another hurdle: how to deliver mRNA to targeted cells. mRNA is notoriously fragile, and enzymes in the human body can break it down.
So you really need to have it protected from the enzymes and other kinds of things in the body. You could call them the bad guys, so to speak. And if you don't, then the therapy won't work at all. It would be non-existent.
As early as the 1970s, chemical engineer Robert Langer and his colleague Judah Folkman proved that it's possible to deliver molecules like RNA inside tiny particles without destroying them. The nanoparticles Langer used were made of polymers, not lipids as used in today's mRNA COVID vaccines.
So we were trying to put DNA and RNA into tiny particles and deliver them. I failed the first 200 times when I did it. And by the way, that was also widely criticized. So I got my first 9 grants turned down. I couldn't get a job in a faculty job in a chemical engineering department, which was my area.
But today, Langer is a renowned scientist with over 900 issued patents. In 2010, he co-founded Moderna and still sits on its board. After decades of development, the main biological roadblocks were cleared. But mRNA technology had never been used outside of clinical trials until recently.
In the last few minutes, we've heard that the first coronavirus vaccine has been approved for use in the UK.
This has been manufactured by the US pharmaceutical company Pfizer and its partner BioNTech.
And here is the first person in line to get it.
In the following months, mRNA vaccines from both Pfizer, BioNTech and Moderna were cleared in multiple countries. Taking less than a year from the lab into people's arms, these are the fastest vaccines that have ever been developed, both with a 95% efficacy rate. But some people are concerned that these vaccines are hastily made.
The speed of making the vaccine, the 10 months that it took, there's very good reasons that people need to understand why that happened. It was an emergency. It was a pandemic. All of the clinical trials were done at the same time. All of the testing was done together. No corners were cut. There was no safety issues that were ignored. Everything was done exactly the same as every other vaccine.
In December 2020, Carico and Weissman were given the COVID vaccines they had contributed to.
And the people were waiting, the healthcare workers in line there to get their vaccine, and they clapped, and then I cried.
About 3.2 billion doses of mRNA COVID vaccines are expected to be given in 2021. And now mRNA may also be used to help fight future pandemics and diseases that current medicines struggle to treat.
The mRNA is just an instruction for the cell to make the protein. We can targeting the messenger RNA to certain organs, certain cell types, bone marrow, and then can perform different kind of treatment.
There are many different applications for messenger RNA. I mean, at Moderna, they're working on heart disease. They're working on personalized cancer vaccines. They're working on rare diseases.
I think the next big act for RNA are probably going to be more vaccines. Last summer, we started working on a pan-coronavirus vaccine. So there have been 3 coronavirus epidemics and pandemics in the past 20 years. You have to assume there's going to be more. Can we make a vaccine that prevents against all of those bat coronaviruses that have the potential to infect people and start another epidemic or pandemic. I think that the pan-influenza and pan-coronavirus are going to be the most exciting RNA vaccines to come out in the future.
mRNA has been proven now as a really impressive and powerful new technology, and the application of mRNA goes, of course, very straight approaches to develop mRNA vaccines against other infectious diseases, and we are already doing that. Yeah, we have started that early on for developing influenza-based mRNA vaccines. We are developing mRNA vaccines against tuberculosis, against HIV.
BioNTech has also been studying the technology in cancer for 2 decades and has a cancer vaccine in phase 2 clinical trials. They're hoping the cancer field might see its first messenger RNA drug approvals in 2 or 3 years. There's no guarantee that other mRNA vaccines and drugs will succeed like they have with COVID-19, but Kariko remains undaunted by the challenge.
That's the most important thing, to accomplish something and meanwhile to be happy. As long as I was in the lab, I had fun. It is just such a joy. Even if things didn't work out how I expected,